All right, we'll continue on with the immune system in this video, looking specifically at humoral immunity, which, if you remember, is part of the adaptive immune response or the specific immune response. First, we're going to review this concept of antigen presenting cells. These are big cells that engulf the antigen, which is usually attached to some sort of cell, present fragments of those antigens to be recognized by T cells and those, remember that antigenic determinant. And these macrophages are antigen presenting cells, typically talking about a macrophage, and I probably will use that interchangeably, usually found in connective tissues and in the skin, because oftentimes that's the first line of defense, and they kind of act as like a mobile sentinels of the boundary tissues. Anywhere that you have a boundary, you can rest assured that you're going to have lots of macrophages there. They basically just eat whatever's there. That phagocytosis, remember that term from probably honors biology, and they enter into the lymphatic system and present that antigen to the T cell in the lymph nodes usually, and then there's an immune response that begins. Um, Macrophages are the best of these APCs, and basically they are the link that that goes between your innate immunity, which is take care of everything, and the specific or adaptive immunity, which is deal with this specific thing. All right, and widely distributed throughout the connective tissues and in the lymph organs. Um, their whole idea is basically uh, once they show that T cell the thing, then they also go out and start eating whatever has the thing. So they become this killer that basically goes around and eats stuff too. They get triggered and go out and attack specific things. So they can be either way, either specific or more general. So a little bit more specifics on the humoral immune response. When you use this word humoral, it just means fluid, typically talking about the blood. And so when you think about something like antibodies, which is this directly responsive with, antibodies are found in the blood almost all the time. Think about antibody tests that people have in conjunction with COVID. The reason that they can do that, or with any other antibody test, is because those antibodies are just found in your blood if you're having an immune response against something. And so basically what goes on, this B cell is going to come in contact with an antigen, and it's going to provoke this humoral response, and antibodies for that specific antigen are then produced. This takes a while to make the exact antibody for that exact antigen, because it could so happen that your body's just never come in contact with that. So it takes a while to get the right thing. And this is why this immune response, the first time in particular, can take some time to occur. These cells then, this is this idea of clone selection, is what this is called, and most of these clones become something called plasma cells. And these plasma cells, or B cells, or this is effector B cells. We'll just call them plasma cells. They are a type of B cell, and they are the ones that are actually producing and secreting the antibodies. They can produce around 2,000 antibodies a second for 4 or 5 days, and then they just die. So that's, if you think about, if you think about protein synthesis and how, it takes us a while to go through that. This is a lot of proteins being produced in a short amount of time, relatively short amount of time. These antibodies circulate through the blood, through the lymphatic system. They bind to those antigens. They basically mark them for destruction, and then the other adaptive mechanisms come by and get them, like the macrophages and whatnot. And some of these clone cells then become memory cells, and memory cells basically are what going to provide this immunological memory. It's a big word there. I have it spelled on the next slide, I'm pretty sure. And so these memory cells kind of remember. And so it could be years later, you come in contact with that same antigen again, those cells are going to still be around, and they're going to remember how to produce that exact antibody. And so then that response is going to be much shorter. So you take something like COVID, for instance, and when you get COVID the first time, it's going to take you a little bit to get over it. If you get it a second time, you may not even recognize that you've had it because your body will act so quickly in response to that. And this is how this secondary immune response typically works. So, a little bit more about this immunological memory. So, there's some more empty space. Got to fill that. That was a good idea again. And so, you have your primary immune response here, usually 5 to 3 to 6 days is when it begins and ends around 10 days is when it's kind of at the most. And so these antibodies are produced for, this is just saying for antigen A. Well, the second time you come in contact with, look how many more antibodies are produced. It's because the factory is already ready to go and it makes all those things. And what this is showing you, if you come in contact with B, there has to be another primary response to that. So every new thing that you come in contact with, you have to kind of set up a response for that. If that makes sense. This lag period, again, for the first time is 3 to 6 days, which is why you kind of experience that more symptoms the first time that you're sick. And whereas when you're re-exposed, it only takes a few hours really, and then after maybe a day, you're already feeling better because this has already been dealt with. All right, and so looking at the two different kinds of humoral immunity that are available to us, there's active and passive. And so go back to English class and think of active and passive verbs. And your English teacher is like an active verb is something you do, passive verb is something that is done to you. And so when you think of humoral immunity, it's the exact same thing. Active immunity is immunity that you are creating yourself. We just talked about naturally acquired active immunity in that you came in contact with a pathogen, your body actively created a response against that, and you dealt with it. This can artificially be done as well through a vaccine. And so basically, what a vaccine does is you are getting those antigenic determinants from the pathogen without getting the actual pathogen. The body recognizes those and goes ahead and informs an immunological response to that. And then you get that without having an active living pathogen in you reproducing. So it's like being able to prepare against the thing without having the actual thing in you. This is why vaccines are so good, is because we get spared the majority of the symptoms of that primary response. And then when we come in contact with the actual thing, it's likely that we won't even know that we had it, which is a good thing. Passive immunity is something that is done for you. And so, how can this happen? Well, it can be naturally acquired. This is the best way of thinking about this is when you are born with immunity from your mother. You're born with some of her antibodies in your blood. And you can keep getting these antibodies through breastfeeding, but whether or not that happens, you still have some of these antibodies in your system. These antibodies don't last forever, only a couple of months, and they begin to degrade. You can also have artificially acquired uh, immunity, and this is when another organism has done it for you, but you receive their immune response against a specific thing. Great example of this is like snake antivenoms, where an animal has formed an immunity against the snake venom, and then you get all the benefits of that without you having to actually do that because, some snakes don't give us a second chance. And so some other animal that doesn't have problems with snakes biting them, usually like large mammals, they form the immunity for us, and then we create what's called like a serum from that. Lastly, talking a lot about antibodies. What is an antibody? Well, an antibody is a protein. Their formal name is immunoglobulins. Immunoglobulin, it's basically just like it sounds, and or IGs. And these are proteins that are secreted by the B cells, and this is basically what they look like. They have this kind of Y shape to them, and they have what are called a light chain and a heavy chain. They have two of these light chains and two of these heavy chains, and they're just called that because The light chain is small, this little piece, and the heavy chain is big. And there are the important part of this is this outside region here. This outside region is known as the variable region of the antibody, which is what gives the immune system its whole efficiency. It's because whereas this whole section is pretty much the same for every antibody, this variable region is going to be different depending on the exact pathogen that you are dealing with. And so the cells these as they make these antibodies, they're basically in a trial and error mode trying to get the right thing that fits the particular antibody. And so then it remembers how to do that. And that's when those memory cells remember how to do it. So the next time you come in contact with it, they can make that exact structure. You've heard me say this 100 times. A protein's shape determines its function. And so you can see how this thing is specifically shaped to basically latch on to some sort of pathogen and just hang on, and this serves then as a flag for the macrophage to come and gobble it up.